the COVID vaccines themselves, if you look at the actual uptake around the world, certainly the United States, it's collapsed, Andrew. I don't think we takes that much more in the sense that very, 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 very few people are still taking these things. And certainly children, the uptake was never very, very high, which is actually really quite a good thing, at least in the United States. So I think that in that sense, the mRNA vaccines for COVID are already in the minds of the public in the marketplace effectively dead. The money that Pfizer is getting from them, the money that Moderna is getting from them has collapsed from the astronomical levels they got in 2021. To me, the next step is the mRNA platform itself. So I've been looking very carefully at the data that have started to come out so the theory, like I'm just going to tell you what the theory is versus the reality. The theory is that I give you this mRNA code, your body, your cells produce the antigen that the scientist, wanted to produce. Your immune system reacts to it, and then trains it to react to that antigen, which is similar to the the COVID virus antigen, right? And that's the theory of the protection. The reality is that the dose of the antigen that's produced, that you're induced by one mRNA strand, um, it could be one, two, three, five. There's no control over it. The dose of the antigen, the dose of the mRNA, you may have some control over, but the dose of the antigen, almost none, because you don't have control over how many copies are made of the antigen per string. The biodistribution of it also is not controllable, right? So you see evidence of harms in lots of different body systems because but one reason might be because the biodistribution of where these antigens are made is not controllable. And the third thing is that the fidelity of the proteins made by the code for the mRNA technology is not perfect. You often get antigens and proteins that are not in the code itself because there's skipping, frame shifts, and other things. And so, from a regulatory perspective, how, as a regulator, do you say, okay, you can give this product to people, even though you don't know the dose that you're giving to people, even though you don't know where it's going in the body, and even though you don't know that you're producing the target antigen that you want to produce? Right. As a future platform, okay, there was a Nobel Prize awarded for the mRNA technology to begin with that allowed it so that it would actually produce the antigens without your body sort of overreacting and with this immune overreaction and destroying it or just getting rid of it. There's going to need to be two or three more Nobel Prize winning advances before this is ready for prime time, I think. So that's the next step is making sure the regulators understand that when they improve products with this mRNA platform, they're approving platforms where the manufacturer has no idea what the dose they're giving, no idea where it goes in the body, and whether the antigen itself that they're supposedly producing is actually produced, and not just off target, and also not off target antigens as well.